The Neanderthals. In fact, some people believe that red hair may actually be a trace of Neanderthal genome that we see today.
Have you ever wondered if tiny pieces of ancient DNA are hiding inside all of us? Scientists have been digging into the genomes of people living today and finding some surprising connections to humans that lived tens of thousands of years ago.
It focuses on a close-knit community of cave dwellers in Siberia who lived 54,000 years ago.
How did these fragments get here? What can they tell us about our past? But what if the DNA scientists uncovered doesn't match Neanderthals, Denisovans, or any known human ancestor we've ever discovered? So who was it? What scientists actually found? Have you heard that parts of very old DNA are still inside people today? Arun Durvasula and Sriram Sankararaman at the University of California, Los Angeles, studied genomes from West African groups, including Yoruba and Mende. Their work showed that some West African genomes carry DNA from an archaic human group for which no fossil genome exists. Researchers describe that source as a ghost archaic population because no matching ancient bone has been sequenced. Their analysis estimated that some West African genomes include roughly 2 to 19% of this deep ancestry.
About 2 to 20% of modern-day West Africans possess this ghost gene.
That range may seem large, yet it points to a substantial legacy of mixing between different human branches long ago. The story moves to high places. Eliecha Huerta Sanchez and collaborators compared DNA from people who live on the Tibetan Plateau to ancient sequences. They linked a particular version of the EPAS, one gene in Tibetan people, to DNA related to Denisovans, an archaic human group first identified from remains in Denisova Cave in Siberia. The EPAS1 variant affects how the body handles low oxygen. People who carry it avoid making too many red blood cells at high altitude. That specific match shows a clear case where DNA from an archaic source contributed a trait that changed how a modern population copes with a harsh environment. Ancient viral traces appear in a different way. Renata Ferreira and Marcelo Briones reviewed old Neanderthal sequencing data and reported short DNA fragments that match viral families such as adenovirus, herpes virus, and papillomavirus. Some fragments came from Neanderthal bone recovered at Chagirskaya Cave in Russia.
The Neanderthals lived in these very caves around 40,000 years ago.
The fragments are short and chemically damaged, but they display patterns that match what happens to DNA after long burial. These viral bits do not mean modern viruses, and ancient ones are identical. They indicate that ancient hominins carried viruses from the same broad families that still circulate today. Recovering such fragments gives a glimpse of infections faced by our relatives tens of thousands of years ago. A third route for ancient viral material is built into our own genome. Human endogenous retroviruses are viral sequences that were inserted into the genomes of distant ancestors millions of years ago. Some of these insertions remain in modern human DNA. Rodrigo Duarte and Timothy Powell at King's College London measured expression of certain endogenous retroviral sequences in human brain tissue. Their work reported associations between the expression of specific viral elements and conditions such as schizophrenia and other brain differences. Those associations are not conclusive proof of cause, yet they show that ancient viral DNA can be active in the body and may influence gene regulation. Major reference genomes anchor many of these comparisons. Researchers at the Max Planck Institute for Evolutionary Anthropology under Svante Pääbo produced the first high-coverage Neanderthal genome.
Compare the Neanderthal genome to present-day people. Neanderthals have actually mixed with the ancestors.
Later, the same institute published Denisovan sequences from remains in Denisova Cave. These ancient reference genomes allowed direct comparisons. They made it possible to identify Denisovan-like matches in modern samples and to trace many non-African genomes to Neanderthal ancestry. The types of ancient DNA that appear in living people fall into three broad categories. The first is direct ancestry from other human groups. For example, many non-African genomes carry Neanderthal segments. Some people in Oceania and parts of Asia carry Denisovan segments. West African genomes show material that traces to the ghost archaic source described earlier. The second category is short viral fragments recovered from old bones and sequencing archives. These fragments record which virus families infected ancient hominins. The third category is endogenous viral elements that integrated into ancestors' genomes and continue to exist in modern human DNA. Every item mentioned involves named people and places. The ghost archaic signal involves Arun Durvasula and Sriram Sankararaman at the University of California, Los Angeles, and modern samples from groups such as Yoruba and Mende. The NORDPASS, one link was reported by Eliezer Huerta Sanchez and colleagues and tied to Denisovan material from Denisova cave in Siberia. Viral fragments were reported by Renata Ferreira and Marcelo Briones from Neanderthal sequence data linked to Chagirskaya cave in Russia. Endogenous retrovirus expression work was reported by Rodrigo Duarte and Timothy Powell at King's College London. The Max Planck Institute for Evolutionary Anthropology and Svante Pääbo appear repeatedly because their ancient genomes provide the benchmarks used across studies. The strength of the evidence varies. The NORDPASS-1 case shows a strong match to Denisovan-like DNA, plus a plausible biological effect on oxygen use at high altitude. The ghost archaic signal in West African genomes rests on statistical approaches that detect deep ancestry inside modern genomes without a matching ancient bone. The viral fragments from Neanderthal data are short and damaged, which means more samples and more analysis will help confirm their exact identity. Endogenous retroviruses are well documented across mammal genomes, yet linking specific expressed elements to health conditions requires further work. These reports together show that modern genomes carry a record of past contacts. Archaic human groups contributed DNA that remains in many populations. Viruses left traces both as short fragments in ancient material and as long-term insertions that became part of the genome. In 2020, Arun Durvasula and Sriram Sankararaman published their analysis and estimated roughly 2 to 19% archaic contribution in some West African genomes. The Neanderthal genome draft was published in 2010 under Svante Pääbo. The NORDPASS, one link to Denisovan-like DNA, appeared in 2014 through work led by Eliezer Huerta Sanchez. More African samples will clarify the story in the coming days. But if our DNA still whispers secrets from ancient beings, what hidden method allowed scientists to uncover these buried genetic echoes? How they found it? So, how did scientists actually pull ancient DNA out of people's bones, or even modern DNA, and figure out that it came from very old human groups or viruses? The process is more than just magic, as it involves careful lab work, computer analysis, and smart statistics. First, they start with samples. For ancient humans like Neanderthals or Denisovans, scientists use bones and teeth from archaeological digs. These often come from important sites such as Denisova cave in Siberia or Chagirskaya cave in Russia. For modern human genomes, researchers use blood or cheek swabs from living populations. For example, West African groups like the Yoruba or Mende, or people from the Tibetan plateau. When they pick which bone to sample, they choose dense parts such as the petrous bone or the inner ear because they trap DNA better over time. Next, the lab work begins. Extracting ancient DNA is tricky. Over thousands of years, DNA breaks down and chemical damage happens. The strands get cut into short pieces and base changes accumulate. One common chemical change is called deamination. That is where cytosine, a DNA letter, loses an amine group and changes to uracil, which the lab machines often read as thymine. This happens especially at the ends of the short fragments. Scientists know that pattern well, and they use it as a signature of true ancient DNA. To deal with that, they run special cleanup steps. They use enzymes like uracil DNA glycosylase and other repair enzymes to remove uracil and repair some of the damaged sites. This makes the DNA more stable and more reliable for sequencing. Without that, you could easily misread ancient chemical damage as a real mutation. Once DNA is extracted and cleaned, they build a sequencing library, basically preparing the fragments so that a sequencing machine can read them. Because the fragments are so short, sometimes just 40 to a few hundred base pairs long, Scientists use methods that favor these short bits. Then they sequence everything. Often they do shotgun sequencing, which reads all bits of DNA randomly, or targeted capture, where they use probes to fish out certain parts of DNA, such as specific human or viral genes. After sequencing, the computer work starts. The short readings from the machine are matched back to a reference genome or viral genomes to see where they belong. For ancient human DNA, they compare it to human reference genomes or to Neanderthal or Denisovan genomes when available. When reading a map, they look at certain patterns to check authenticity. For example, they check for the damage signature. Those cytosine to thymine changes are especially near the ends. They also check how long the fragments are. Ancient ones are often shorter than contaminant DNA from living people. But what about the strange ghost archaic DNA that Durvasula and Sankararaman reported, without any reference to fossil DNA? Here is the clever part. They used a statistical method called a hidden Markov model, HMM. This helps them to scan across the genome of modern people and mark out segments that look very different, as if they came from a deeply diverged branch. The HMM basically treats each region of DNA as archaic or modern and compares how dense the unique variants are, especially those not seen in outgroup populations. That way, they can spot archaic ancestry even if they do not have DNA from that archaic group. Another method is S', which looks for archaic variants in modern human genomes. It pulls out segments that likely came from Neanderthals or Denisovans by comparing variation patterns. This helps confirm introgression events in people. While they map DNA fragments and run these statistical scans, scientists also run contamination checks. Ancient bone samples might be contaminated by DNA from microbes, or even from people handling the bones. To guard against this, labs use special clean rooms, full-body protective gear, and strict protocols. They run negative controls, samples known to be blank, alongside the real samples to check for rogue modern DNA. They also estimate contamination using mitochondrial DNA, the small circular DNA in cells, because it is abundant. If the mitochondrial genome in the bone sample looks more like present-day humans than ancient humans, that signals contamination. They also compare damage patterns. Modern contaminant DNA tends to have long, undamaged fragments, while ancient DNA is shorter and damaged in ways that match what we expect after thousands of years. When researchers search for viral sequences, they take a similar route. Suppose they look into old Neanderthal sequencing data. They compare the short readings against a database of viral genomes, such as adenovirus, herpes virus, and papillomavirus. When they find a match, they check for mapping quality. They ensure the fragment truly aligns well to that viral genome, and that it does not align just as well to something else. Then they check for the ancient damage patterns on those viral readings. Is the fragment short? Does it show cytosine deamination? And is its length consistent with being very old? If it checks out, that suggests the virus was present in the ancient hominin. Because viral DNA in bone is usually rare, scientists repeat the analysis in multiple bone samples or sequencing runs. If they see viral fragments in more than one sample, that strengthens the argument that these were real infections long ago, not just lab contamination. For the human endogenous retroviruses, HERVs that live inside our own genome, they work a bit differently. They look at modern human tissue, like brain tissue, and run RNA sequencing to see which viral-like sequences are active. If a HERV sequence is being transcribed, it means the viral fragment inside our genome is not just dead junk. It might be doing something. Researchers compare expression levels across individuals and between people with different conditions, for example, neurological differences, to see if there is a relationship. Throughout all of this, statistics are everywhere. Scientists run simulations to test how likely a pattern is to appear by random chance. They check alternate explanations, such as an error in sequencing, population structure, or contamination, before concluding they really have a piece of archaic DNA or a viral fragment. When possible, they run the same analysis in different labs. Reproducing results adds strength to their claims. To give a real example, when Durvasula and Sankararaman mapped segments in West African genomes, their hidden Markov model also estimated when that archaic ancestor split from modern humans, and roughly how much of the genome came from that branch. Those estimates are based on models of population history, recombination, and mutation rate. In another case, when researchers looked at Neanderthal bone data for viral fragments, they reran mapping and damage checks to rule out contamination and confirm they were seeing ancient viral DNA. There is also a more recent advance. Scientists are now exploring how to read chemical marks on ancient DNA, such as methylation, a kind of chemical tag on DNA bases. One new method measures methylation directly and more safely than older approaches. That work is still early, but it may let scientists reconstruct not just the sequence of ancient DNA, but also some of the chemical modifications those ancient cells used, giving more insight into how their DNA was regulated when they were alive. Every step is done carefully. From choosing the right bone, to extracting fragments gently, to sequencing and analyzing the data using smart statistical tools, the scientists leave little to chance. Their goal is to tell a trustworthy story about the past, based on real DNA. This is how they go from a broken, damaged piece of bone in a cave to recognizing a snippet of viral history from tens of thousands of years ago, or spotting DNA from a human group we do not yet have in any fossil. It is difficult work, but when done right, it reveals our deep past that was once thought lost forever. But if these fragile clues can rewrite everything we thought we knew about our origins, what deeper secrets are still hiding in our DNA? What this means? The reports reviewed earlier show patterns about human history and genetics. First, humans often mixed with other human groups. This mixing left traces of DNA that persist today and can affect traits in modern populations. The NORDPASS 1 gene in Tibetans provides a concrete example. People living on the Tibetan plateau carry a version of this gene that traces back to Denisovan-like ancestry. This version affects red blood cell production and helps the body handle low oxygen at high altitude. Researchers observed that this gene variant became more common in Tibetan groups over time because it provided a survival advantage. This is a direct case where archaic DNA influenced a trait still present in people today. Other examples focus on DNA inherited from Neanderthals outside Africa. Many people carry segments of Neanderthal DNA in small amounts. Studies have shown that some of these segments can influence immune system function and skin characteristics. The effect varies across populations, and the exact impact is still being studied. These findings demonstrate that mixing with archaic humans contributed to genetic diversity, creating variation that may have had practical effects on health and adaptation. The second pattern involves viruses. Ancient hominins carried viruses that left traces in their DNA. Analysis of Neanderthal bone samples, such as those from Chagirskaya Cave in Russia, revealed small fragments of viral DNA. These fragments match known viral families, including adenovirus and herpes virus. While the viruses were not necessarily identical to modern strains, these findings show that some infections humans face today have very deep roots. Recovering viral fragments allows scientists to study the history of infections and how they may have influenced human evolution. A third source comes from human endogenous retroviruses. These are viral sequences that were inserted into the genome of distant ancestors millions of years ago. Progressively, some HER-Vs became part of normal mammalian biology. One well-studied example is the gene syncytin, derived from a retrovirus, which plays a role in forming the placenta. Researchers at King's College London studied HER-V expression in human brain tissue and noted potential links to conditions like schizophrenia and amyotrophic lateral sclerosis. These links are not proven causes, but they show that viral sequences retained in our genome can influence gene activity and may have functional roles in tissues today. Despite these insights, there are still questions that scientists aim to answer. The identity of the archaic ghost group in Africa remains unknown because no ancient genome matches the signals detected in modern populations. Researchers hope new samples or improved laboratory methods will clarify the origin of this DNA. Another open question is the timing of these mixing events. Some occurred tens of thousands of years ago, while others may have been more recent. Better dating of samples and alignment with archaeological evidence will help refine these timelines. Preservation is another challenge. DNA in African climates often degrades quickly, reducing the number of ancient samples available for study. Cold, dry regions like Siberia preserve DNA more effectively, which is why much of our information about Neanderthals and Denisovans comes from these locations. Scientists continue to develop techniques to recover and analyze DNA from less ideal samples. From a health perspective, these findings show that archaic DNA can influence disease risk and response to infections. Certain gene variants from archaic humans affect immune system responses, though the impact can differ across populations. HER-V sequences may also play a role in gene regulation and health, but further research is needed to understand their exact contributions. These results highlight a combination of past human interactions, viral history, and retained genetic material. They provide evidence of how our genomes reflect both ancient mixing and long-term viral influences. Each observation is supported by laboratory work and statistical analysis, helping scientists build a detailed picture of human ancestry while leaving open questions for future exploration. Thanks for watching. If you enjoyed this video, hit like, leave a comment, and subscribe for more stories. Share your thoughts on which part fascinated you the most.